This section explains the single idea that organizes the whole first chapter of biochemistry. There is no vital force. Life is inanimate, molecules obeying exactly the physics and chemistry of dead matter. Every property that looks like an exception, self-maintenance, self-replication, sensing, evolution, is a composition of ordinary interactions, with no new law required. The chapter lays 4 axes: cellular, chemical, physical, and genetic, from which the entire book is derived. It crowns them with evolution as the single principle explaining both unity and diversity, the same monomers, the same code, one ancestry, endlessly varied. Whole degenerative move, given only conservation of ordinary chemistry plus heritable variation, the boundary between living and non-living dissolves into a question of organization. We are made of stardust and nothing more is needed. This section explains why life needs no law beyond ordinary physics. Life is inanimate chemistry, running a program that copies itself. Every biomolecule, examined alone, obeys the same physical law as a rock. The surprise is that no extra law is needed: complexity, ordered structure, and self-perpetuation. Ordered structure and self-perpetuation all emerge from ordinary molecules coupled through energy flux. That coupling is the step that does the work. An organism spends energy from its surroundings to hold itself away from equilibrium. The state matter drifts toward when left alone. The chapter's 5 foundations, cellular, chemical, physical, genetic, and evolutionary, are 5 faces of that one primitive. Predict the rest from it. Anything alive must eat, meaning take in energy, must remember, meaning carry genes, and must drift, meaning evolve, because those are what a self-copying program against entropy requires. This section explains how water, the solvent, acts as a constraint that decides in advance which biological structures can exist. Everything a cell builds, water-shaped first. The solvent is not a stage. It is a constraint that pre-selects which structures can exist. One trick generates the whole chapter. Water's bent, polar geometry lets each molecule hydrogen bond. 4 neighbors, so it clings to itself. Anything that can join that network, ions and sugars, dissolves. Anything that cannot, oils and lipid tails, gets excluded. They are not pushed out by repulsion. They are squeezed together because water maximizes its own disorder around them. That single piece of entropic bookkeeping is the hydrophobic effect. It is what folds proteins, closes membranes, and pairs the double helix. Read water structure, and you can predict biomolecular architecture without touching the biomolecules. The medium writes the form. This section explains what Chapter 2 installs before any biochemistry proper begins. Life's architecture is not chosen by the molecules. It is imposed on them by the solvent they sit in, and that solvent is water. Water is a player in the chemistry, an active participant rather than a passive stage. Its molecules hydrogen bond to themselves so cohesively that everything dissolved in it must negotiate with that cohesion. Polar solutes join the bonding. Non-polar ones are expelled and clumped together, not from any mutual attraction between them, but because water reorders itself to exclude, and clump together, but because water reorders itself to exclude them. So folding, binding, recognition, the whole 3-dimensional order of proteins, nucleic acids, and membranes falls out of a single competition: solute against solvent, set against solvent, against solvent, against solvent. Individually negligible weak forces decide structure only because the medium sums them all. To predict any biomolecule's shape, ask what arrangement leaves water's own bonding least disturbed. This section explains why a protein reduces to a single sequence. There are 20 monomers, one shared backbone, and a single point of variation, the side chain. Everything a protein is gets written in the order of those side chains along one directional strand. The peptide bond forms by condensation. A carboxyl meets an amino group, water leaves, and the reaction is intrinsically ordered. That ordering gives the chain an N to C polarity, like text read one way. So, the entire molecular repertoire of life collapses to a combinatorial primitive, a small alphabet linearly sequenced. Diversity comes from arrangement. The parts stay simple, and the order of them is where the variety lives. This is the same move as language, and as the genetic code. Fix the sequence, and you have, in principle, fixed the protein. This is why the chapter treats primary structure as the master variable. This section lays out the structural primitive that proteins are built on. 20 side chains, one linear thread, and from that almost nothing comes every function a cell performs. The chapter's thesis is a combinatorial alphabet. Fix a small set of monomers whose R groups are each chemically individuated, concatenate them in sequence, and the space of orderings, 20 to the nth power, already contains enzymes, antibodies, keratin, luciferase. The generative primitive is the same one that runs DNA and written language, the alphabet in which the language of protein structure is written. But here each letter carries its own charge, size, and solubility, so linear order alone dictates the fold and thus the function. Arrangement is where the diversity comes from. The parts themselves carry none of it. Everything downstream, structure, catalysis, regulation, is this one move, elaborated. This section lays out the single organizing idea behind protein structure. A one-dimensional sequence names a point in a space of astronomically many conformations, yet the chain collapses to essentially one shape, the native fold. That collapse is the chapter's one, primitive. Sequence selects structure. Structure licenses. Function and folding is free energy minimization, the chain settling into its lowest state rather than being assembled piece by piece. The part that should update your model is this. The fold is held together by a swarm of weak interactions, each one individually near zero, and not by strong covalent bonds. The dominant contributor is the hydrophobic effect. Burying oily side chains releases ordered water, and it is the water's entropy gain that pays for folding, not any bond itself. Stability is only marginal, 20 to 65 kilojoules per mole. So the native state is a shallow basin, dynamic and sometimes partly disordered. That malleability is itself often the function. This section explains the single primitive that governs protein structure. A protein's amino acid sequence encodes not the maximally stable shape, but a marginally stable one. The native conformation sits only 20 to 65 kilojoules per mole below the unfolded state. That thin margin is the near cancellation of two large opposing tendencies. On one side, conformational entropy and solvent hydrogen bonding pull the chain apart. On the other, weak non-covalent interactions, the hydrophobic effect foremost, pull it together. Countless theoretically possible conformations collapse to a handful that predominate. The reason is that function demands a structure poised to change, to bind, to catalyze, to unfold, not one locked rigid. That is the whole chapter's primitive. Order in proteins is the small residue of two large numbers, and everything downstream, the four structural levels, denaturation, folding, is bookkeeping on that razor-thin margin. This section explains what protein function reduces to at its most basic level. Everything a protein does reduces to one move. It binds a ligand reversibly at a site whose shape and chemistry are complementary to the partner. transport, catalysis, immunity, motion, signaling, each is that single act plus a twist. Binding deforms the protein, and the deformation is coupled to work. The surprise the chapter builds toward is that affinity need not be fixed. Hemoglobin's 4 sites talk to each other, so oxygen binding at one raises affinity at the next. This is cooperativity, bending a lazy hyperbola into a switch. Regulation, then, is nothing new. It is modulated binding. Grasp reversible complementary binding plus conformational coupling, and you can predict the next protein before the chapter names it. This section explains why binding alone, stripped of catalysis, is already a complete form of protein function. Strip catalysis away and binding alone is already function. That is the chapter's whole wager. A protein does work on the world without changing the molecule it touches. It just holds it reversibly, then lets go. Reversible complementary binding coupled to conformational change is the generative primitive. Specificity comes from shape and charge fit. Action comes from the induced fit, the binding triggers. Every case is the same program at different scale. Hemoglobin carrying oxygen, an antibody recognizing an antigen, myosin walking along actin. Enzymes in chapter 6 are only this primitive, plus one more step. Transform what you bound. So the chapter isolates the binding half of protein function before catalysis complicates it. This section explains the single idea behind enzymes. Catalysis, not replication, is the quieter of life's 2 conditions. A bag of sugar sits inert on the shelf for years, yet your body burns it in seconds. The whole difference is an enzyme, and its whole trick is one move. The enzyme does not grip the substrate in its stable resting form. It is shaped to fit the transition state, the fleeting, high-energy contortion a molecule must pass through to react at all. Binding that stabilizes that peak lowers the activation barrier, written delta G double dagger, and the rate rises exponentially. Everything downstream, kinetics, mechanism, regulation, elaborizes exponentially. The surprise that reorganizes your whole picture is Haldane's. Catalytic power lives in weak bonds to the shape. A molecule almost never holds the transition state, not the shape it rests in. This section explains catalysis, the second of the 2 things life needs. Life needs 2 things: to copy itself and to hurry reactions it cannot afford to wait for. This chapter is the second half, catalysis. The generative move is Haldane's. An enzyme spends its binding energy not on holding the substrate, but on gripping the fleeting transition state more tightly, collapsing the activation barrier, delta G double dagger, while leaving the activation barrier, delta G double dagger, while leaving the equilibrium untouched. Everything downstream is that single asymmetry unfolded, specificity, saturation kinetics, every named mechanism, the regulatory switches. To predict the next enzyme, ask only what its pocket complements, the ground state or the transition state. The answer is always the transition state. That is why it catalyzes at all. This section explains why carbohydrates are the cell's densest information carrier. Fuel and scaffold are the alibi. The chapter's real claim is that sugars carry more information per unit than any other biological polymer. Here is the generative primitive. A monomer's message capacity scales with its degrees of connective freedom, not with its count. Each glycosidic bond chooses a linkage position, an anomeric face, either alpha or beta, and whether to branch. So, oligosaccharides span a tree of possibilities, where nucleotides and amino acids only span a tree of possibilities, where nucleotides and amino acids only span a chain. Two glucoses join one way to make a fuel store and another way to make cellulose. Identical residues, divergent worlds. From that combinatorial surplus follows the whole payload. Glycoconjugates act as address labels, the sugar code read at cell surfaces for recognition. Structure is the message. This section shows why sugars carry more information per residue than any other biopolymer. Every biopolymer is an alphabet, and its power is just the number of distinguishable arrangements it admits. This chapter exists to show that sugars win that count. A nucleotide or an amino acid joins its neighbor one way in a line. A monosaccharide carries several hydroxyls and a chiral anomeric carbon, so two identical glucoses can link at different positions in alpha or beta, branched or linear. Same residue, combinatorially more words. That is the thesis, and it threads from stereochemistry through glycosidic linkage to the glycoconjugate surface. Beyond their roles as fuel and scaffold, carbohydrates are the third informational polymer, the sugar code, and the densest one. This holds precisely because branching time stereochemistry beats a linear four-letter chain per unit length. This section explains what a nucleotide is and why its structure is the whole reason heredity has a chemistry. A nucleotide is three parts snapped together, a base, a sugar, and a phosphate. The chapter's whole thesis is that once you can polymerize such a monomer through a uniform phosphodiester backbone, the only thing that varies along the chain is which base sits where. So the molecule becomes a one-dimensional sequence, and sequence is information. Here is the step everything depends on. The bases pair complementarily, A to T, G to C, so each strand specifies its partner exactly. That single fact, a linear polymer that templates its own copy, is why heredity has a chemistry. Everything downstream, replication, transcription, and the genetic code is bookkeeping on this one structural primitive. The same nucleotides also moonlight as energy currency and as signal. This part explains one molecule wearing many hats and why a single one of them holds the others. A nucleotide is a base plus a pentose plus a phosphate, and that same molecule is metabolic cash, the ATP that pays for cellular work. It is the messenger in a cell's response to hormones. It is the scaffold of enzyme cofactors, and it is the letter of heredity. The chapter's thesis is that this last role subsumes the rest. Line up four bases, and their sequence specifies every protein, every RNA, and ultimately every biomolecule the cell builds. Life compresses to a one-dimensional text over a four-symbol alphabet, copied faithfully across generations. The primitive to carry forward is this. Arbitrary biological form is the readout of a linear code. Structure is downstream of sequence. Everything after this chapter, the helix, replication, sequencing, is machinery for storing, reading, and copying that one string. This section explains the toolkit that turns DNA from something you read into something you can act on. Sequence is information, but the previous chapters claim only bytes once you can act on it. This chapter is the toolkit that turns DNA into an addressable, editable substrate, and every tool in it is one primitive in different clothes. Complementary base pairing, the same rule that stores and copies biological information, is also the handle that lets you find a sequence. Copy it, cut it, and splice it. Consider the four tools in turn. Restriction enzymes. Recognize a specific word. PCR exploits templating to amplify. Sanger sequencing reads by complementary synthesis. Cloning recombines fragments into a vector. Reading and writing collapse into one mechanism. Now predict the rest. Any manipulation you want reduces to arranging where complementarity is allowed to act.